Thank you for the kind introduction. So, yeah, I hope everybody's having an awesome conference. I'm going to talk a little bit about the world's largest biomedical data set, which is the UK Biobank. Then I'll talk a little bit about how regular bioinformaticians can really analyze that data in sort of an multi-omic way. So, how many of you would call yourselves bioinformaticians of some kind, data scientists? All right, so about 2/3 of the audience, so that's pretty cool. The rest of you just buckle up, try to follow along. I got 49 slides in 25 minutes, so hopefully that'll work. So how many of you know what the UK Biobank is? Okay, almost everybody. So the UK Biobank, for those of you who don't know, is 500,000 people. Study started 14 years ago. Now there's whole genome sequencing for all of those people, whole exome sequencing for all of those people, blood biochemistry, full electronic medical records. And then eventually, right now, there's proteomics for about 10%. That'll be on everybody. And then now there's also a single cell data set being launched. And anybody can get access to this data set. So if you are interested in access, you can go ahead and apply. I work for a place called DNA Nexus. DNA Nexus is a platform to do massive scale bioinformatics on the cloud. And we host the UK Biobank as well as a bunch of other biobanks, including Our Future Health, which will be 5 million people with EMRs and SNP chips, as well as the Mexico City perspective study and others. So that's pretty cool. And I really enjoy working in the biobank segment. If any of the grad students have questions about working in industry, please feel free to talk to me afterwards. I'd be happy to talk to you. So yeah, I mean, so basically when people get UKB access, so access to the UK Biobank, and by the way, if you're an early career researcher, i.e. graduate student or postdoc, even if your lab is not interested, you can get access very, very inexpensively, and your PI can still pay for that through their grant. So works out pretty well. Whenever anybody gets access, they get automatic access to the UKB wrap, which is the system that sits on DNA Nexus. And I just want to show you what one of these systems looks like. And my big motivation for that is because for human data anyway, we'll probably see more and more of this type of data sitting in these enclaves. Other examples are all of us, FinGen, those types of things, where data really can't be moved. Instead, researcher access is granted to people doing legitimate research who are not going to violate the terms of any particular agreement. So really, I want to show you UK Biobank really is a representative example of what a data enclave can look like. So, and one thing I want to point out here is that, so at the DNA Nexus anyway, everything that you see on the GUI, you can do on the command line as well. And that's something that I find particularly convenient, being sort of a command line person myself. But anyway, there's a basic file system. You can think of that as sort of an Amazon S3 bucket over here. You can monitor your jobs, so on and so forth. You have access controls, so you can add people as long as they're on UK Biobank and run this kind of concepts of apps and workflows. I think apps are really important as we move into sort of large-scale computing, really on big data sets, because we can have encapsulated pieces of compute and transfer those around. So if you think of sort of Docker within a system, and we'll talk a little bit more about Docker later. But I've been a computational biologist for maybe 15 years now, and one of the things I've always wanted to be able to do is slice and dice data. So here I can take all females between the ages of, I don't know what I put there, 45 and 60, who have breast cancer and take cod liver oil, right? And I can slice that out of 500,000 people in just a few seconds. Also, personally, as an American, I find cod liver oil fascinating because it's like a thing we tell our children to give them nightmares, whereas in like the UK, 5% of people in UK Biobank take cod liver oil. So that's an actual thing. So anyway, so that to me is awesome because I can slice those and then jump into the VCFs or whatever, grab that data, jump into the proteomics data, grab that data, as I'll show you in a few minutes. And the really nice thing here is I can use Jupyter, RStudio, if that's something you like. I can use Spark Jupyter Lab if I really need to move a lot of data into the Jupyter environment, and also these encapsulated apps. One thing I will say, though, is at least on the DNA Nexus enclave and many of these other enclaves, you know, I mean, it's really nice to use encapsulated apps and workflows when you're dealing with really large scale data instead of an interactive interface like Jupyter or basic command line. And so that's a really nice thing. And, that the nice thing about sort of the DNA Nexus setup, although other places have similar things, is you can grab anything you want from GitHub or Docker, build it into one of these apps, and then you can build things into workflows. So DNA Nexus supports WDL and Nextflow, as well as to some extent CWL. And so those things can be compiled into DNA Nexus workflows. If you're a bioinformatician and not familiar with Docker, WDL, Nextflow, probably something to check out. These kinds of interfaces are really growing in our field, and that's a pretty big thing. But another nice thing for people that aren't really sort of super savvy in that way is we make it really easy to do GWAS. So as long as you can think rationally about covariates, it's pretty trivial to do GWAS. As I showed you before, you can slice and dice on, women with breast cancer between 45 and 60.
Excuse me?
I can take
just the slides are not moving.
Cohorts and compare them. That's a relatively easy thing to do. So
sorry about that.
No problem.
Go ahead.
Thank you, Seth. Anyway, so and lots of people like to do PheWAS, which is basically the opposite of a GWAS, right? So you take a SNP, you think about which phenotypes are associated with it. But as I'll talk about a little bit later, I think we need to move from individual SNPs really to haplotypes. But here's the really mind-blowing thing. Not only do we have... Slides don't want to advance. Give me just a sec. I'll fix it. I got it. So not only do we have genomic data, we have proteomic data, as I mentioned before, which is super cool and something I'm very excited about, but also imaging. So we have 40 million images. I gave an education talk a couple of nights ago talking about learning imaging, right? And teaching a bunch of genomics people how to do basic imaging, which has been quite an adventure for all of us, I think. But it's really cool now that we have, for all of those people with breast cancer, I also have an abdominal MRI, right? So I can look at images of these folks, and then I can take predisposing SNPs or haplotypes or whatever, and use imaging as a longitudinal readout to see how they're doing, how they're responding to certain drugs, that kind of thing. So that's kind of mind-blowing. By the way, we also have a bunch of metabolomics data. I know some of you are interested in that. Finally, if you're a sort of regular bioinformatician, you can grab a cloud workstation or TTYD, basically just command line and online, which is really nice. And then you can build your own Git, Docker, that kind of thing. Finally, I want to talk a little bit before I sort of go off the rails and talk about crazy things about longitudinal phenotypes of medical records. So this is all harmonized to OMOP. So we can cross between different data sets. So for example, if you're an academic researcher, you can get access to UK Biobank, analyze on that, and then cross what you're doing to all of us, right? Now, pro tip, it's about a third of the price to operate on UK Biobank versus all of us just because of contractual arrangements with cloud providers. So AWS, which hosts UK Biobank, gives about a 70% discount. So it's about the cheapest commercial cloud you can get to. And then finally, the really cool thing, we can think about multimodal data analysis, integrating imaging, as well as, somewhat federated analysis. Although I want to be very clear, we're not porting all of us data over into the DNA Nexus system and vice versa. But what we can do is port compute across. So say I'm working on UK Biobank and I'm working on all of us. The really cool thing is I can use Docker to move code from one to another. And that's really helpful. And then I can wrap the Docker with Nextflow or WDL or whatever. But out of curiosity, of the graduate students in the room, how many of you know how to use Docker? Awesome. All right. That is a great proportion. I was at a different institution on the other side of this state, and the proportion was way lower, which makes me very proud to live in Western Pennsylvania. But anyway, if you don't know how to use Docker yet, here's a couple Docker tutorials that we did with HDR UK. So that's super cool. And finally, for those of you interested in the machine learning side, one thing we did, this was with my colleague Junaid Nashad, who worked in my group and is now finishing a PhD at Oxford University. We took electrocardiogram data and we were able to slice and dice it, predict the features that were most likely leading to arrhythmias, and then put that back into this sliceable, diceable stuff. So really, this gives you some sort of a platform where really meaningfully, sort of machine learning researchers can interface with clinicians. And I think that's really very cool. Clinicians as well as epidemiologists. And I think that's really cool. So that's sort of, I guess, the overview of sort of the world's largest biomedical data set and its hosting by DNA Nexus. And that's a pretty exciting thing. But now I want to sort of go off the rails and talk a little bit about oncological subgroups and really some of the things we can do with these large data sets to really change the way we think about medicine. And that's what I'm really sort of passionate about. So, I think it's really interesting to think about colorectal cancer. And there's this thing we know about colorectal cancer, that it's actually not one disease, right? It's one ICD-9 code, it's one ICD-10 code, but it's actually 4 distinct diseases. And this is from the Guinea et al. Paper, Nature Medicine, 1996, right? 2016. And what we can see here is that we have 4 diseases which represent different percentages of colorectal cancer. But what they don't necessarily mention is that if you treat CMS1 people like CMS type 4, they actually die faster than if you hadn't treated them. So this really has major impact. So it's a work with Emerson Hewitt and Andre Kubica. We looked at some of these germline somatic mutations versus somatic mutations and tried to come up with predictors of what drugs would work for various subtypes given particular driver mutations. And that's something that, I'm very excited about and passionate about. But I think, again, we have to go beyond just thinking about SNPs because we have enhancers that interact with the coding regions, but also there's a lot of copy number and structural variants. So, and I think that's a pretty big deal. And we can use graph genomes really to integrate these types of things. And I'll show you some examples of that in about 5 minutes. But the other really cool thing we can do is integrate proteomic data, right? So I think proteomic data is really exciting because, the UK Biobank has proteomic data and, this whole genome data. And unlike RNA-seq data, proteomic data is kind of your one-stop shop for absolute expression, right? There's no translational noise, any of that, et cetera, et cetera. And so the first project that I was involved in with proteomic data was with Scott Beeler and Duke Tran in Kelly Bolton's lab at WashU. And we looked at progression towards myeloid neoplasms based on proteomic data. And really, to make a long story short, the paper for this is out. But anyway, we were able to predict subgroups of myeloid neoplasms and also transition to myeloid neoplasms based on proteomic data from the UK Biobank. And so, I'm going to skip this slide. So then thinking about this, one thing we'd really like to do is coming back to graph genomes, we'd like to take the whole graph and really think about returning haplotypes for subpopulations. So, and we were able to do that reasonably well with individual SNPs previously and do some prediction. But we weren't able to actually integrate that with proteomic data. And this is really a sad note. So for the UK Biobank, the proteomic serum collection actually happened after, or the proteomic blood collection actually happened after people were enrolled. In fact, 10 years after people were enrolled. And the sad thing about that is anybody who came in with malignant colorectal cancer was no longer around to give blood. So with Scott Beeler's advice, we actually switched over to prostate cancer. And I'd like to talk about this subset of prostate cancer, which is actually particularly aggressive. And that's really interesting because all the men in this room, if we lived to be 200, every single one of us would get prostate cancer. but only 10% of that prostate cancer would actually be really sort of malignant and invasive. About 90% of it would be benign. And we only have like clues to why, we only really have clues to why some of that is malignant. But one of the signatures for malignancy seems to be an upregulation of SPINK1 and a downregulation of CHD1. And so that's something we wanted to look at because a lot of that comes from like Western blots. We don't really know what's going on. So, and here you can see that SPINK1 is involved in aggressive cancer. But anyway, so I was able to slice and dice this in the UK Biobank, right? So basically I go to C61, which is malignant prostate cancer, the ICD-10 code, and I say, well, I just want all protein signatures. So these are proteomic signatures for SPINK1, and I want all of the ones that are above 0. So that's a really nice and easy thing to do. But here's a really nice thing. Then what I can do is I can go and look at CHD1, right? So the story is CHD1, when it's down, SPINK1 is up, and then you get aggressive prostate cancer, right? So this would be really nice to know. And here's the really mind-blowing thing. Those mutations in SPINK1 are probably germline. I'll show you the evidence in a minute. But here's the cooler thing. We're talking about the largest biomedical data set in the world, right? So you'd think, oh, I need huge compute, and I need to have an enormous amount of knowledge. How many of you can write a one-liner bash script? Great. I'm going to show. How many of you can do a blast search? Everybody should be raising their hand. All right. So I'm going to show you that with 3 one-liner bash scripts and a blast search, you find out everything you want to know about the relationship between CHD1 and SPINK1 in this. So here, all I got to do is go into this huge 500,000 person VCF. And I'm looking, actually here, I'm looking at the phase data for 200,000 people. But what I can do is just run BCF tools, right? So I just take the ones with SPINK1 overexpressed, and I'm just going to grab the CHD1 locus from there. So that's pretty cool. And then what I can do is I can build FASTAs from that, and I can build graphs. And why am I building graphs? I'm building graphs just to check that there's no super major rearrangements, et cetera, right? And so, and then I can put all that, applet, Docker, so on and so forth. In fact, we did that. I'll show you a couple of interesting things we saw. So in CHD1, we see, granted, there's no depth here, so there could be additional copies, but we see no major rearrangements, changes, all that. I'll show you a control for that in a little bit. But what we do see is that there seem to be about twice as many variants in the SPINK1 overexpressed and SPINK1 underexpressed. But it's not premature stop codons. We used OpenCravat, which is a really easy to use annotation tool to check for that. So there don't seem to be any premature stop codons. So what we theorize is actually that you have differences upstream, probably in the enhancer regions, and a number of SNPs that are probably related to aggressive cancer. Now, I told you I'd show you that in these genome graphs, you can see rearrangements. So here's a bunch of clusters of a different gene, which is called TAC, which is will drive malignant prostate cancer even farther, but is not necessary for malignant prostate cancer. And here you can see you have 4 or 5 clusters of things upstream of TAC. But going back to CHD1, really what we wanted to do was make clusters of CHD1. So this is where the blast search comes in, right? So what if I take all of these haplotypes for CHD1, and I mean, since I have no rearrangements, they're probably inherited together. And remember, this is phase data, so this is diploid. And then what if I blast one of these against the rest of them? I just make a blast database, make blast db -nu db type nucl. Anyway, so here I can make that. And what I can do is I can blast one of these. Whoops. I can blast one of these into the rest of them, right? So that's pretty cool. And then I can get these sort of clusters of what goes together. And it's very hard to see here, I'm sure. But basically, we're seeing most of the diploid pairs come together. I cut off the EIDs just because of like privacy concerns for UK Biobank. going to give a little bit of this talk at ESHG, and they'll be there. So just to make absolutely sure it's private. But then what I want to do is I want to put together sort of the phenotype network with the genotype network, right? And so I want to put those two things together, and I want to say, oh, okay, how do I overlay, this sort of ad hoc, very simple bioinformatics haplotype network with my phenotype network? So all I have to do is label the ones of these that are associated with SPINK1 overexpression, right? I don't have to do anything super fancy. I don't need fancy diagrams, because if I label them, check this out. So those are the ones with SPINK1 overexpression, right? So basically what we see is that SPINK1 overexpression is correlated with these similarities between CHD1 haplotypes. But the possibility of this is mind-blowing, because if this is all correct, and there's a lot of work to do still here, but if this is correct, what that means is that there's germline mutations here, where if somebody gets a driver mutation for prostate cancer, and all of us will at some point, this may mean that we have germline haplotypes that will actually direct that prostate cancer to be aggressive. And we're all walking around with that, which has implications for cancer screening in the clinic. So I think that's a pretty cool story. And so what we're doing now is we're looking for the marker SNPs for these haplotypes, and then we want to go look across other cancers to see if some of these germline haplotypes are predisposing for other types of aggressive cancer, because that obviously has even more implications in the clinic. And so that's a pretty cool thing, I believe. And so then to recap, we can think about, which combinations of loci, et cetera, and then eventually we'd like to think about which drugs work most effectively for these people. But again, I think the screening implications are pretty huge here. So for the next 2 minutes, we go through real quick a little bit of work we've done with transformer models and why that matters to what we're doing here. So I talked to a lot of people about transformer models because, DNA Nexus supports a lot of commercial companies and their cloud adventures and so on and so forth. Interesting, we saw a lot of people were very interested in validators. That was super cool. And we thought a lot about what you need to set up an ethical transformer model, but really a lot of it is clean data. And that's a pretty big deal. And I think the biggest impact we've seen from transformer models over the past year in biomedicine is that people are actually cleaning and harmonizing their data, which make other models work way better. I saw some great talks yesterday where they were like, look, Lasso works amazingly well now. And I'm like, that's because somebody cleaned the data, right? Like, so that's kind of awesome. Like, people all of a sudden care about ontologies. I mean, people are talking about RDF all over the place. Like, man, it was like 100 people in Japan and Chris Mungle were all waving the RDF flag for 10 years. And like, nobody would listen to them. And now, like transformer models are a thing and everybody's back to RDF. So if you don't know what those things mean, just ask me later. So anyway, so I'm kind of trying to nudge DNA Nexus to do a lot more of this stuff. And so here's a bunch of stuff, like out there in the community that's like super cool for loading. And the upshot of this is that really, knowledge graphs are awesome for loading. But one thing we didn't know how to do is load VCFs. And so my friend Chao Feng Lin worked with a bunch of people in Pittsburgh and figured out how to load VCFs. And this is all on GitHub. So if you want to try loading VCFs into transformer models, you should, because it's fun. But then we also realized it's kind of expensive, right? Like to load like big knowledge graphs and big VCFs into transformer models. So we thought about that a lot. And so my friend Emerson Hewitt, along with a bunch of brilliant people from Pitt and CMU and my buddy David Anoma in Nigeria, figured out a way to subset knowledge graphs for RAG. Because actually, if you use a huge knowledge graph for RAG, it turns out to be really, really expensive. And I can tell you that for those of you who are graduate students and postdocs, you will anger your PI if you spend like $25,000 on trying to run transformer models. They will be unhappy with you. So please don't do that. So anyway, so I'm really excited about the potential that transformer models have for things like optimizing these haplotype phenotype associations and really routing different data to different models, right? Because we can call lots of different models with transformer models. We can call simpler models and so on and so forth. We could probably save everything in some sort of concatenated knowledge graph structure. So a lot of people did a lot of work on these various projects over the past year. I've highlighted a whole bunch of them, thanks really to Xiaofeng Lin and Scott Beeler and Andre Kubica. I think I have 2 or 3 minutes for questions. So if anybody has a question, I'm happy to answer them. Hi.
Excellent talk. I have a question about some of the clinical data that is provided. I know you mentioned gender, age, and also like the ICD-10. How in depth does the ICD-10 go, I guess? Because I know for like thyroid cancer, it's C73, but sometimes like with the thyroid nodules, depends on like the size of your treatment. Is that some stuff that's offered, like the stage of cancer and things like that, or even like the treatment that they're on?
So I guess here's how I might phrase it. So there's 7,000 phenotypic fields. So there's a lot there, and some of it is more up to date than others. So in terms of treatment, you should be able to get all the treatments for every patient, right? Because but let me be clear, that's for hospital interactions. It's all coded. For primary care interactions, we do have treatment. But it's not coded at that level, for example. Stage is usually annotated, but this kind of subtyping, like I'm showing with colorectal cancer, is not, right? So, and I think that's something that, sort of we as researchers have a responsibility to think about, like how to subtype these large databases and return the data to UK Biobank so they can theoretically publish this and say, for other researchers. And that's something I didn't mention, but UK Biobank has a mechanism where you do a large-scale study, you can return it to them and they can make that public. So that's probably the next step there. But there's, there is an enormous amount of data on cancer. One thing that's interesting, the cancer death registry is actually a couple years behind just because of political reasons. So it turns out that all the hospitals in the UK have to report cancer deaths to the central government, and then that comes back to UK Biobank. But which is an efficient system, it's just time lag. So it's about 2 years behind. But typically with late stage cancer patients, if they disappear and they're no longer going to the hospital, that you can make an inference on what happened to them. And if you use those inferences, your numbers tend to line up.